All right, so introduction to the genetics of Alzheimer's disease. As you sadly probably know, Alzheimer's disease is a very prevalent neurodegenerative condition that goes through a series of stages. We first have, I'm going to zoom in here, but we first have mild stages in which there is some memory loss, some speech difficulties and mood changes. I learned recently that there's also some, and I say these with an agenda of sorts, there is also some sleep disorders associated with the early stages of Alzheimer's disease. So sleep disorders are now being used among some of the signs that there may be an onset of the condition. Then there is a other stage with learning impairment. So now it's not about remembering stuff anymore. It's about not being able to learn new stuff. Aggression, dementia, and impaired daily activities. Some of these aggression, by the way, is thought to be due to those sleep impairments that happen associated with the condition as well. So it's almost like, people who suffer these moderate stages of the condition are constantly sleep deprived in a way, and that doesn't help. And then, of course, the severe stage when patients are bedridden and there is major motor impairment, right? There is two forms of Alzheimer's disease, and this is something that from a genetics perspective applies to all kinds of diseases, except maybe for infectious ones, right? So any other condition that is not result of some sort of infectious or pathogenic infection will have, generally speaking, two forms. The familial form, right? That is due to some sort of genetic inheritance, and we call it familial form because it happens within families, and you can follow the condition in family trees. And then the sporadic, right? So you'll hear this terminology when it relates to classifying conditions in human health very often. So we can speak of familial and sporadic Alzheimer's disease, familial and sporadic types of cancer, familial and sporadic diabetes, and so forth, right? It's simply the fact that sometimes the condition manifests within families with an obvious pattern of inheritance, and sometimes it shows up in families and in pedigrees, quote unquote, unannounced, right, of sorts. So that's why we call it sporadic, because it's not happening with a clear inheritance pattern. Is just showing up in a family. So one distinction between the familial form and the sporadic form of AD is the familial form tends to be early onset. And as I said before, it presents with a clear genetic component because you can see that the condition shows up in earlier members of families. The typical age of onset for the familial form is around 40 years of age, whereas the sporadic, it's a little later, and this is the one that, like I was saying before, sadly, most of us may be familiar with, is the onset happens around age of 65 or older. Right? And there's no clear genetic component in that there is an individual who's experiencing late onset Alzheimer's disease, and then you ask about their family history, and no one had it in the family, so there's no clear familial component. And that is not to say, and this is something that I should kind of take a step back, this is not so much about modern molecular genetics, but it is definitely about the genetics of health and disease. When we say that a condition is sporadic or that it doesn't have a familial inheritance pattern, that is not to say that condition won't have any genetic basis or any genetic component, right? So often we make a distinction between genetic diseases, those that we can put our finger on a given genetic cause for the disease. That is not to say that those diseases for which we can't do that, there is no genetic component at all. And in fact, late onset Alzheimer's disease is no different. Many years ago, and to this day, because these studies continue, they're constantly on an ongoing basis, people took thousands of individuals who had not experienced late onset Alzheimer's and thousands of individuals who had, then compared their genomic sequences and conducted what is called a GWAS. And a GWAS stands for Genome-Wide Association Study. And essentially what it is comparing the genomic sequences of a very large number of individuals with a condition versus the genomic sequences of a very large number of individuals without the condition, and figuring out if there is any pattern of correlation between genetic markers in one group versus the other. And doing that, identified a series of genes that are not normally associated with the early onset Alzheimer's disease, but that were still statistically more prevalent among those who experienced late onset Alzheimer's versus those who didn't. And those genes are not super strongly associated with the development of the condition. So it is thought, or in those cases, it is concluded that those loci predispose carriers to developing the condition, right? So there is that interaction between carrying alleles that are associated with a given condition and certain environmental factors that may make someone develop, in this case, Alzheimer's, but in general, the condition. Yeah? You had a question, Lara?
Yeah, I'm at 25.
Oh, absolutely. 40 is most likely an average. Yeah. So when people say that the typical age of onset is 40, that is most likely representative of an average or a median, some sort of statistics of centrality. But I bet that it can set on much earlier. Yeah, there may be cases.
I read some article that women tend to start Alzheimer's symptoms
Earlier. So the comment here is that women may start experiencing symptoms earlier. I had heard that as well. How do you guys explain that?
Hormonal imbalance, maybe?
It could be hormonal. Yeah, in general, we can say hormonal. It could also be certain environmental factors that are associated more typically with women in humans. So it's hard sometimes to distinguish between the two, but at least it's a starting point for saying, all right, now I can look into these much more laser focused, in a much more laser focused manner. Yeah, there was a question in the back? No?
You said it. I was just going to ask if it could be like X-linked, like the disease for...
Excellent. So the common X-linked, if it could be X-linked, depending on the type of allele associated with even this late onset, if it is X-linked, in fact, you might expect that it manifests in males more quickly because they have only one X. So if they're carriers of that predisposing locus or loci, they have no other copy back up. Whereas females in the human population will have two, and often what happens is the one carrying the faulty locus gets silenced and the other one is fine. So you would expect the opposite. But that is not to say that there is in other aspects of sex-linked genetics that have to do with biological sex and a series of secondary characteristics that occur during development. Yeah.
One of the...
There you go. There you have it. So that's a beautiful example of what I just said, in that higher or elevated levels of FSH are a secondary characteristic that is not necessarily related to the X chromosome, right? It's just that throughout development there is a wiring of the hormonal axis such that females may express higher levels of FSH. Particularly, I dare say, particularly at the middle age, where you might start seeing the bifurcation between males and females in terms of incidence of AD. Good. All right. I didn't use any images from outside of the 5 reviews that I shared with you. So if any of these images look familiar, that's why. I took them from those articles. This is a pretty cool diagram that kind of shows on the left what a healthy brain looks like, and some of the concepts to bear in mind are that in healthy brains you have healthy mitochondria instead of neurons and healthy microtubules. That will become apparent why that is important in a second. And neurons are in a way devoid of any depositions of insoluble proteins around them. And there is a healthy microglia and a healthy BBB, that stands for blood-brain barrier, meaning there is a pretty tight control of what goes into the brain and what comes out of the brain and into the bloodstream. In an advanced Alzheimer's brain, there is a reactive microglia, which we normally refer to collectively as neuroinflammation. So there's a whole bunch of chemical reactions that are happening within that brain that have to do with increased blood flow, division, a whole bunch of chemical reactions that will generate reactive oxygen species and so forth, oxidative radicals and so forth. From the point of view of the neurons are damaged, particularly at their termini, so where they make connections with other neurons. And if we were to look inside of them, you'll see that there is mitochondrial dysfunction and there is dysfunction of the microtubules. The microtubules are not moving things in and out of axons as they should. And yes, of course, outside of those neurons, you would probably find the amyloid beta plaques, and inside of the neurons, the neurofibrillary tangles. The reason I think the interesting reason why this cartoon emphasizes mitochondria and microtubules is because for a really long time, people thought that the culprits of Alzheimer's disease were these amyloid beta plaques that existed between neurons in the extracellular space that were basically, I heard terminology many years ago like choking the neurons and not letting them do their job, hence the memory impairment, hence the learning and cognitive impairments, right? Now the thinking has changed, and the thinking is that now it's more of the damage that is happening inside of the neurons that often precedes what happens outside of them, and that in a way explains the slow progression of the disease before you can see any of these massive amyloid beta plaques. Okay. Did we say anything? Everything. Oh, and the last but not least, again, for a long time you would hear, that the most severe forms of Alzheimer's were correlated and therefore people assumed due to the deposition of these amyloid beta plaques and the tau neurofibrillary tangles, right? So most, if not all of the reviews that I share with you will say something along the lines of Alzheimer's disease is pathologically defined by two major lesions: the extracellular deposits of amyloid beta plaques and the intracellular or intraneuronal neurofibrillary tangles. Yeah? Any questions, anyone? All right. So, as in a way I was saying, now we have some graphics to back it up. For the longest time, people would in autopsy would dissect the brain out of severe Alzheimer's patients and see these beta amyloid plaques and the tau tangles, and they thought, well, this is it, right? This is what is causing the disease. But then, as people studied where and how to look a little better, and these studies were refined, and then people started looking at people who had passed without having developed the more severe forms of Alzheimer's, but instead showing some of the milder ones, they could start drawing some tighter correlations between the appearance of these plaques and tangles and the development of the cognitive disabilities, right? And what that showed is that the total AV plaque burden correlated only weakly with development of the disease, whereas neuronal loss and the neurofibrillary tangles correlated more strongly, right? So faced with that evidence, I come here and I tell you the plaques correlate weakly with disease progression, the NFTs correlate much more strongly. What would you conclude drives the progression of AD? The amyloid beta hypothesis. We'll get there. We'll have to refine it though, and that's the key. Josh, what were you gonna say? Do you remember what that gene is by any chance? APO4, right? Yeah. So APO4, we'll put a pin on that. But I'd like to go back to this question, faced with that better evidence that plaques don't correlate tightly with disease progression and NFTs do, what's the most natural conclusion? That NFTs drive the thing, right? So there was a period that the focus changed from the plaques, which were the ones that were found first, to the NFTs, because with better evidence, the evidence now was saying, well, hold on a second, we see the plaques everywhere in super advanced Alzheimer's patients. But in earlier stages of the disease, where we can clinically start seeing some of the early signs of Alzheimer's, we see no plaques, but we start seeing some NFTs. So maybe all this time we put the cart in front of the horse, right? And maybe it wasn't that the plaques were causing NFTs or that they were happening somehow simultaneously. Maybe it's the other way around, that NFTs are causing the plaques, right? And are the true drivers of the condition. So if that is the case, right, what mutations would you expect to be associated with the familial form of the disease? That is a way to say, what mutations would you expect to almost invariably cause early Alzheimer's? Gabby?
I read the APP, the amyloid precursor protein, which before the abnormal cleavage produces those amyloid beta, like oligomers, aggregates, which are toxic to neurons.
So you share the punchline with us. But that's where I wanted to get. If we go with Arcia's idea that NFTs are driving the thing, then you would expect that mutations in genes related to NFTs, tau protein itself, or any of the enzymes that can drive the formation of these NFTs, would be invariably linked to the progression of the disease. But that's not what people were seeing. What people were seeing whenever they went back to those families with Alzheimer's disease, a history of early Alzheimer's, they kept finding mutations in those genes, APP, presenilin-1, presenilin-2, that had to do with the formation of the plaques.
Yeah, something I was looking into earlier too is I thought maybe a mutation in kinases that were hyperphosphorylating the tau, but I didn't see anything on that either.
That's right, and what you did is what many researchers did for many years. Once this tighter correlation between NFT formation and disease progression was established, they thought, well, let's look for mutations in the genes that contribute to the NFT formation, because reason has it that if those genes are mutated, someone will develop AD. But they couldn't find those mutations, right? You can always find them, but they were not associated with development of the disease. So that forced everybody to take a step back and rethink the whole thing, right? And start looking a little more closely, because there was some sort of disconnect here between the epidemiology of the condition and what we knew about the genetics of the condition. So the epidemiology was saying, it seems like you don't need the plaques to develop AD. There is no tight correlation between plaque formation and disease progression. It kind of seems like it's NFTs that you need to drive the disease. But on the other hand, we know that those who develop the disease invariably and early have mutations in the beta plaque pathway or beta plaque formation pathway. So there was a disconnect there, right? So the solution came when research showed that way before the beta amyloid plaques are formed, there is an accumulation of these amyloid beta oligomers, right? So it wasn't so much the plaques, but these AB oligomers that represent the most neurotoxic species. That sort of changed the whole tune. For years, people thought these massive plaques around neurons, they must be, as I was saying before, half jokingly, they must be choking the neurons. But then research showed that no, there were these amyloid beta oligomers that were poisoning neurons way before there would be any formation of beta plaques. And that those amyloid beta oligomers were actually the ones that trigger the cascade of events that lead to tau protein hyperphosphorylation and entanglement. Right? So I have some text here from the paper. Soluble amyloid beta oligomers accumulate at synapses, where neurons communicate with one another, where they disrupt calcium signaling by messing up the NMDA receptors. And what happens is I had to do a little a little deeper digging in the literature. But in a nutshell, what happens is these amyloid beta oligomers will, in a way, hyperactivate NMDA receptors. So you will find a lot of references in the literature saying that the oligomers affect or disrupt NMDA signaling. When we say disrupt, we tend to think that it will kill it or inhibit it or break it. It's actually the opposite. It's disrupting in the sense that it's making it hyperactive. And with hyperactive NMDA activity, there is a sudden influx of calcium in the neurons, inside of the neurons. That influx of calcium has been shown activates these two kinases, CDK5 and GSK3 beta, both of which have plenty of substrates in cells. They participate in a whole bunch of pathways, but both of which can phosphorylate tau at various positions, right? And it is the hyperphosphorylation of tau that will lead in turn to the formation of the NFTs. Not drawn there, but implied. Conversely, and this is one of those things that is probably the evolutionary wiring of neuron biology made it such that so let me put it this way. Evolutionary wiring of this network made it such that NMDA is, on the one hand, controlling calcium influx and indirectly these kinases, but NMDA signaling is also controlling this phosphatase inside of cells, inside of neurons, called PP2, stands for protein phosphatase 2, in a way that NMDA, when active, inhibits the activity of PP2, right? So it's a double whammy here because the hyperactivation of NMDA is on the one hand inducing a calcium influx, which will indirectly hyperphosphorylate tau, and on the other hand, it is inhibiting the activity of phosphatase 2, PP2, that normally dephosphorylates tau. So now poor tau is being hyperphosphorylated on one side and is no longer being dephosphorylated by PP2. So it's a compounding effect, and now there is hyperphosphorylation of tau, which leads to the formation of the NFTs. And all of this is triggered by these amyloid beta oligomers present in synapses that will go and physically mess with NMDA receptors and lead to their hyperactivation. Okay? So now we can put it all together. The story is a little more complicated than everybody thought, right? So it all starts with these amyloid beta oligomers that will, on the one hand, eventually lead to the formation of plaques, but much sooner than that start messing around with synaptic activity and namely lead to the hyperphosphorylation and entanglement of tau. And because tau is normally associated with neuronal and axonal microtubules that are important for what a neuron does, which is communicate with other neurons. For neurons to communicate with other neurons, they need to bring a whole bunch of neurotransmitters to the end of axons, and they need to bring back any waste or recycling material that they need to send back to the soma, and for that they use the microtubules. You mess with that, you mess essentially with the transport system, so now a neuron will soon be left without any neurotransmitters to communicate with the next one in the chain of communication, right? And it will start accumulating toxic waste as well. So far, so good? All right, cool. Now, that is not the only thing that amyloid, I'm going to get it right at the end of the semester, amyloid beta oligomers do. They also serve as a signal for microglia to start secreting cytokines that induce inflammation, local inflammation. So microglia, along with other glial cell types, in the context of Alzheimer's, the other important one are astrocytes. Microglia and astrocytes are the cells in the brain that are not neurons. I don't know if you guys had heard of these before, probably, that the human brain has really two kinds of cell types. There is the neurons doing all the communication, the wiring, and information processing, and then there is these support cells that we collectively define as the glial cells. Some of them have true supportive function, as in they will bring nutrients for the cells and provide growth factors and protect them from damage. Some of them are there to patrol and make sure that they deal with any infection or injury, like the microglia. Some of them are there to, I don't know if you guys ever heard this, but wrap around axons and make sure that the conduction of the electrical signals through neurons is much faster. These are the Schwann cells and myelination, right? So all of these cells that have nothing to do with electrical signaling inside of the brain, but serve as a support system. So research showed that these amyloid beta oligomers can trigger pro-inflammatory responses in microglia. And these microglia will start producing all of these inflammatory cytokines that will dilate the blood vessels, bring in more immune cells that will start secreting things that they produce to kill pathogens, but end up killing host cells. It's a mess, right? Like any inflammatory process that it was evolutionarily meant to fix some damage and be resolved, but if it is sustained in time, because there is no pathogen, but there is a lot of those oligomers that fool the microglia to think that there is some sort of infection, now this prolonged inflammation ends up having a ton of secondary effects, right? From a genetic network perspective, one of the aspects that your reviews emphasize is that the oligomers can activate these microglia by binding to this receptor in the microglia called TREM2. Okay, so several of the reviews mention TREM2 as the main partner of oligomers waking up microglia and inducing the expression of inflammatory cytokines. Did I say everything? Yeah, there is an aspect that I didn't include in this cartoon, and that is that the oligomers outside of neurons can attract microglia, right, and wake them up. but also oligomers inside of neurons doing, at synaptic connections, doing all that hyperexcitation of the NMDA pathway and all these calcium influx and all of that will lead to axonal and synaptic injury. And when an axon or a synaptic button are injured, they will secrete a bunch of danger or alarm signals that will as well recruit the microglia and activate them. So it's not like microglia is activated only by the TREM2 receptor binding to the oligomers. It can be recruited simultaneously by signals, damage signals coming from the neurons themselves. Okay? What else? So last and perhaps not least, and this brings us back to why I was saying I think it's important how that cartoon of the brain emphasized mitochondrial function. This is relatively new, and when I say new, maybe 30 years old, but in research years is pretty new. Research show that these oligomers can mess up with the function of mitochondria. And as much as you remember your mitochondrial biology, mitochondria, the powerhouse of the cell, basically takes nutrients, turns them into ATP. When it works, great. When it doesn't work so well, two things happen. There is less energy around for the cell to operate with, and there is an increase in the production of these reactive oxygen species or free radicals that will themselves cause damage in other cellular components. So these reactive oxygen species can go and cause mutations in DNA. They can go and mess up with proteins and so forth, right? So these oligomers of the amyloid beta peptide will insert themselves between mitochondrial membranes, mess up with the electron transport chain. Basically, they go and mess with mitochondria. And now it has become apparent that damage to neuronal mitochondria way precedes whatever damage having a massive plaque outside of the neuron could do to neuronal function. Now we know a little better, and we know that the neuron is malfunctioning way before there is a gigantic plaque sitting right next to it. Okay? Cool. So, with the reactive oxygen species and this sort of crazy environment inside of the neurons, that happens to, in fact, induce further oligomerization of any free amyloid beta peptides that may be floating around. So it's sort of a vicious circle that worsens the problem. All right, so I mentioned now a couple of times the amyloid beta peptide, right? Because the oligomers are the ones that cause trouble, but they come from oligomerization of peptides that are, we refer to them as monomers, right? Short peptides. So to have oligomers, you first need to have the monomers. And where are those monomers coming from? Well, it turns out that they come from a type of cleavage of a membrane protein that mostly neurons express in the brain, but glial cells will as well, called APP. And APP stands for amyloid precursor protein, right? Amyloid precursor protein. So APP, as shown here in this cartoon, let's go with what happens normally first. So APP is a protein that is expressed in the membrane. There is these enzymes that can cleave the extracellular domain, and they do so sequentially. So in this case here, you see first cleavage by alpha secretase, and then whatever is left is cleaved further by gamma secretase. There may be combinations of base 1 cleavage followed by gamma secretase cleavage, or base 2 cleavage followed by gamma secretase cleavage. All of these numbers here refer to where in the APP protein the cleavage takes place. And depending on where the cleavage takes place, where the next cleavage might occur as well. And as you can see, there is a whole bunch of options, and there is several proteases that can cleave APP. What matters in the context of AD progression is that not all of the peptides that are produced by cleavage of APP are toxic. Not all of them are going to be these famous amyloid beta monomers or peptides that will in turn oligomerize and form the amyloid beta oligomers, right? So down here, there is the combination of cleavages that do no harm. But then up here, there is the two major combinations that do cause the production of amyloid beta monomers or peptides. And in a way, in both cases, it all comes down to the sequential activity of an enzyme called base 1 followed by gamma secretase in a specific combination of amino acids. I showed you down here there's an exception to that. So there is instances in which base 1 and gamma secretase can work together and do no damage, but there is these two other possibilities in which they will produce the amyloid beta peptide. And that amyloid beta peptide that they produce comes in two flavors or forms. There is the 40 amino acids long peptide and the 42. And you probably read among your reviews that the more 42 species there is present in an organism, the higher the chances that they will start oligomerizing and causing trouble. And in fact, it was recently approved as a diagnostic tool to measure the ratio of 42 to 40 in blood. So you don't have to go poking inside of the brain, in anyone's brain. Simply take a blood sample. Of course, so far it's not for just anyone. It's for people with other risk factors, right? But one of the diagnostic tools that is going to be used now is they will take blood, they will measure the ratio of 42 to 40, and there's going to be some tables that say high risk or low risk. Okay. Any questions, anyone? So biochemically, you have an APP protein that is sitting in the membrane and that is normally cleaved into smaller fragments. Sometimes it's cleaved into smaller fragments that do no harm, but then sometimes there is this amyloidogenic pathway that will generate the amyloid beta peptides that can oligomerize and start causing trouble, right? And what I did is, from a genetic perspective, I sort of drew this little cartoon down here. Zoom in on it. The idea here is that if we think of the amyloid beta oligomer as the end product that will in turn trigger the progression of the condition, there is these genes that in a way contribute positively to the accumulation of the amyloid beta oligomer. There is APP itself, because the more APP cells express, the higher the chances that they're going to be cleaved through the amyloidogenic pathway. And there is, of course, the two main suspects or main culprits of generating the oligomers, that is gamma secretase and base 1. And the reason I'm emphasizing it this way is because these are going to be nodes that we can later explore by ways of asking, how does dopaminergic signaling affect the expression of APP? Or how does sleep disorders affect the expression of base 1? So that's how we will enter the Alzheimer's disease network through these important genes. Ashia, you have a question?
Yes, so APP is like a tree that like chop off.
More than a tree, like think of it as a trunk, right? Because the peptides are linear in a way. And think of a trunk that maybe we chop off the top first and then the bottom. But think of it as more like a light pole rather than a tree. No branches in it. Most proteins, unless those that are decorated with sugars, most proteins are just a linear sequence of amino acids, right? So think of it as a light pole, and you don't want to chop it down all at once. So you go some way, halfway up, truncate some, and then cut the rest. Does that make sense?
So no branches. Right?
No branches, yeah. So if you look at all these cartoons, these are cartoons, but you never see any branches in these cartoons. It's like one long stick, if you will. Now, that is not to say that the protein will not fold in ways that end up having some sort of 3D shape that may look like branches. But from a molecular continuity point of view, there is no branching. It's just one molecule. Yeah? Likewise, and again, thinking about the amyloid beta oligomers being the kickstarting point for AD, one can say that alpha secretase and base 2 will inhibit the formation of those. Why? Because it turns out that base 2 and alpha secretase cleave APP in a manner that won't generate any of the amyloid beta peptides that will oligomerize. Right? So in a way, the more base 2 and the more alpha secretase there is in a neuron, the higher the chances that there will be any production of the amyloid beta peptides that are needed for the formation of the oligomers. Right? So from a regulatory perspective, we're saying that these two inhibit amyloid beta oligomers. Now, I need to be very explicit about this. I'm not saying that APP, gamma secretase, or base 1 activate the oligomers in a sense of doing something to the oligomers to make them more potent in any way. What I'm saying is that these three are contributing to the formation of the oligomers, and these two contribute to lessening the formation of these oligomers. Right? So that's the mapping from a regulatory perspective. These two nodes inhibit that one, and these three stimulate it. Yeah? I'm going to share this with you for what it is. In doing my reading, I read that the APP gene is located in chromosome 21. Is chromosome 21 familiar to any of you for other reasons? Trisomy of 21. So what is the condition that it causes? Down syndrome, right? So Down syndrome is a condition that happens when an individual has 3 copies of chromosome 21. And all of the pathologies and all of the phenotypes associated with Down syndrome are thought to be due to the fact that there is that extra copy, and therefore more of the genes that are encoded by chromosome 21, which will cause some sort of genetic imbalance leading to the phenotypes associated with Down syndrome. So I didn't know this, but I'm like, well, hold on a second. So if APP is in chromosome 21, right, and more APP means a higher chance that you're going to be generating more of the oligomers, one might expect that people who suffer of Down syndrome may have a higher incidence of Alzheimer's disease. And sure enough, yeah, they do. So this idea that the more APP you have, the more oligomers you might have, pans out at least from that point of view. That for individuals who suffer of trisomy of 21, they will have more APP expression and some sort of like correlated or collinear accumulation of oligomers earlier on. And there is higher incidence and younger age of onset for Down syndrome patients. Yeah. Wonderful question. Wonderful question. So I think I'm going to bring it back to what Gabby was saying earlier, that one of the genes that was first known to be associated with the familial form of the disease was APP, right? So you're asking, well, the APP gene is there already. So what kind of mutation is making this APP, or what kind of mutation could make this APP more prone to generating AD, right?
Right, but when we were talking about the fact that it's like the way that it's cut, right?
Yeah.
That's like determining whether or not the APP is turning into the alpha or the beta form of.
Correct. So I think you have it. Let's unpack it together. So the way that it's cut determines whether or not you're going to form an A beta peptide, 40 or 42, or not. So what kind of mutation could tilt the balance towards formation of the 40 or 42?
Person is on.
Cool. So what Ali is saying is, for instance, if I make a mutation in AEP and I somehow
Now,
like
we're making AEP or have that present in the body.
present more or something like that, I'm going to have more of this cleavage and therefore more of the A beta. Is that what you're saying?
Yeah.
All right. What you just said, totally true for gamma secretase. So there are mutations associated with familial disease in these two genes called presenilin 1 and presenilin 2. Those two live here. They're components of the gamma secretase complex. So what you just said, well, maybe I can make a mutation, or there could be a mutation that makes this happen more, is exactly what happens with the presenilin 1 and 2 mutations. Now, since Gabi brought it up, we know that one of the mutations that is associated with familial form are mutations in APP. So let me rephrase it. And the question goes for everybody. Let's say that I came to you and I said, can you please make me an APP, a mutant APP, that won't go through the non-amyloidogenic, but will always go through the amyloidogenic pathway? What would you do? What mutation could you make in APP such that it always goes through the upper pathway and not the lower one? That's it. That's it. So what we need to do is, where is it? So basically, and I don't know if this is the case, right? But conceptually, if you mutated this lysine 17, which is the site of cleavage for alpha secretase, you're basically killing this. And now you might make this APP more prone. You've, in a way, increase the chances that it goes through either of these two amyloidogenic pathways by about a fifth, right? So before it had 5 different pathways, now it has 4 because you kill the place for alpha secretase cleavage. So that's how mutate. Yes, APP. Everybody has the APP gene, but those carrying certain mutations of the APP gene or presenilin 2, 1 or 2, will favor the formation of these oligomers. Did that make sense? All right. So let's move along. We just discussed how these monomers are produced. That the monomers themselves, they're not really doing anything bad. It is the oligomers that are bad news, right? So monomers are apparently produced with a certain probability all the time because these pathways that we just discussed coexist. It's just a matter of who bumped into that APP, if it's an alpha secretase or gamma secretase, base 1 or base 2, how that APP will be cleaved. Monomers are produced all the time. Oligomers are bad news. So what can organisms do, and what do organisms do to prevent oligomerization? So what would you do to prevent oligomerization? I'm looking for like general ideas here. I don't need mechanisms, names. So if you have monomers that can turn into oligomers, how can you prevent the formation of oligomers? So reduce the APP, and in a way, indirectly, I suppose, reduce the monomers, right? Reduce the monomers. And Josh, you were going to say something?
I guess we reduce the factors that make them adhere to each other.
Reduce oligomerization. That's exactly right. So if you want to prevent the formation of oligomers, either you prevent the process itself or you take out the substrate for that process, right? And that's exactly what organisms naturally do, right? Some of it has to do with preventing oligomerization, but much of it has to do with getting rid of these A-beta peptides, the monomers. So there's a series of mechanisms that are related to reducing the number of monomers present in the brain. One is direct enzymatic degradation. So there is all of these enzymes, I listed them there, neprilysin 1 and 2, endothelin converting enzymes 1 and 2, angiotensin converting enzyme, and one that we'll hear about later today, matrix metalloproteases, so MMPs. All of these are enzymes that, for the most part, not always, but for the most part, live outside of cells, and they're there in a way with a defensive purpose or a defensive function, just cleaning things that might be getting too close for comfort to a cell. And those effectively can take these amyloid beta monomers and chop them up. And making those monomers disappear will alleviate the problem. Another way that is now getting into some of the more complex genetics, if you will, is those monomers that exist outside of cells. Remember that the APP is a membrane protein whose extracellular domain and part of the transmembrane domain are cleaved. So all of this stuff ends up on the outside of the cell, right? So one of the things that brains do is they say, let's take it back up. Let's take it back up, send them to lysosomes, these recycling plants of cells, break them up into amino acids and reuse those amino acids. Let's recycle the amino acids, right? So that's exactly what happens. So there is endocytosis and degradation via lysosome, and this happens in all kinds of brain cells, neuron astrocytes and microglia, and that's where our friend ApoE first comes into place, right? Or in the picture. So it turns out that there is this protein called ApoE. Your reviews make a lot of emphasis about how they come in 3 flavors. 2 of them are non-harmful, but one of them, ApoE4, was associated through those GWAS studies to late onset disease. So someone carrying the ApoE4 allele, if they carry 1 copy, they have an X percentage higher chance of developing Alzheimer's. If they have 2 copies, if they're homozygote for it, they have an even greater chance of developing late onset Alzheimer's. But even the non-pathogenic alleles, E3 and E2, are present, and they're actually helping out. They're clearing these monomers that are present in the extracellular space. And the way they do it is they're produced by the astrocytes. The ApoE proteins have 2 binding domains. One of them binds lipids, and the other domain they have binds to receptors in cells. So in this case, they will bind the amyloid beta monomer and bring it to one type of receptor called the LDL receptor. In this case, in a microglial cell, but it could be any other cell that expresses that receptor, and helps the amyloid beta monomer go into the cell. And once inside of the cell, the fate of this amyloid beta monomer is typically go to a lysosome where it will be degraded into its component amino acids, right? So it's a relatively straightforward story. There is this thing lurking around in the extracellular space, and there is ApoE3 and E2 that come, pick it up, scoop it up, and send it inside of cells where it can be degraded and recycled. I had a question for you. So how can I mentioned that the ApoE proteins have a lipid binding domain and a receptor binding domain, and all they're doing is bringing the lipid to the receptor so that they can go inside of the cell. How can a lipid binding domain bind a peptide, the amyloid beta peptide?
Well, I think I read that these peptides, they have like a hydrophobic section.
Uh-huh. Did everybody get that these peptides have a hydrophobic nature? That's right. If we go to the previous, I'm not going to do it, but if we go back a couple slides, we'll see that the A-beta peptide comes from inside of the phospholipid bilayer. It's from the transmembrane domain of APP. So we can tell biochemically those peptides are going to be highly hydrophobic. So for all intents and purposes, to ApoE, they may look like a lipid. ApoE's other jobs have to do with carrying lipids. They will carry cholesterol, they will carry other lipids, and send them back into cells, because a lipid that is floating around in extracellular space is useless. So ApoE and a family of apolipoproteins are there to shuttle back lipids into cells. And ApoE happens to have the ability to bind the amyloid beta peptide because of this hydrophobic nature that it has. Right? Cool. So let's move on. Then the other thing, and it turns out that this is the major mechanism for clearing the brain up of any A beta monomers, is through transcytosis through the, it got off there, transcytosis through the BBB, through the blood-brain barrier. And the way this works is very similar to what I just described, except that the ApoE bound to the A beta monomer will bring the A beta monomer to a receptor expressed by endothelial cells, blood vessel cells in the brain. And this receptor is called LRP1. Same thing, the ApoE and amyloid beta monomer are going to go inside of the cell through endocytosis, but this time, instead of being shuttled into a lysosome for degradation, they will be sent to the other end of the same cell and expelled into the blood vessel lumen, into the bloodstream through exocytosis. So that's what transcytosis means. There is an endocytosis event, that's right here. So there's an endocytosis event on one end of the cell. In the reviews, you'll see that it's called the abluminal end. Then there is a vesicle that is formed, and that vesicle will go to the other end of the cell and undergo exocytosis. So whatever came in through endocytosis will be transported through the cell and then dumped out on the other end of the cell, which happens to be the blood. And the blood will take that amyloid beta monomer and take it out of the brain and all the way into the liver or kidney, and there dispose it. So this is the mechanism that will take care of approximately 40 to 60% of the amyloid beta peptides, for the 40 and the 42, that are produced in the brain. And this is what we are now using for early diagnosis. So whenever we find that an individual is carrying too much 42 or a too high a 42 to 40 ratio in blood, it's a good indication that we don't know yet, but in the brain there will be formation of oligomers soon, toxic oligomers that may kickstart AD progression. So once again, from a cartoon point of view, you okay? Yeah. From a cartoon point of view, what I'm saying here is that you will have fewer oligomers if you have a working ApoE and a working LRP, because working LRP and ApoE should reduce the amount of monomers by 40 to 60%, both of which will reduce by 40-60% the formation of the oligomers, right? Cool. There is a kink to this story, and that is that ApoE in any of its isoforms, 2, 3, or 4, can become lipidated. Remember when at the very end of Monday's lecture, I mentioned that one of the post-translational modifications that proteins can experience in cells is the addition of either palmitoyl fatty acids or myristoyl fatty acids. So something like that can happen to ApoE. It becomes lipidated, meaning that there is a fatty acid that is added to its structure by a covalent bond. That's the job of these lipidases called ABCA1 and G1. And once ApoE has this hydrophobic tail, Now, lipidated ApoE proteins will come together and form these micelles because those tails don't like to be surrounded by water, right? So as they start bumping into one another, they will want to stay together and they will form these lipid droplets. It's almost like these were phospholipids, except that it's a micelle that is happening with the ApoE proteins on the outer shell and all of these lipid tails inside. From a dynamic point of view, what that does is it keeps all of these ApoE proteins together, makes them unable to easily find the LDL receptor or the LRP receptor, but they're still able to bind the A beta monomer. So they're still carrying cargo, but they cannot so easily dispose it where they should. In fact, there is a shuttling of these lipidated ApoE droplets to a different kind of receptor that can kind of dispose of some of the A beta, but much more slowly. Right? So, all of this to say that lipidation of ApoE means that the discharge into the blood vessel through the BBB will be reduced. Okay? From a network's perspective, ABCA1 will inhibit free ApoE, will in turn induce the formation of lipidated ApoE, and lipidated ApoE prevents the recycling of A beta, and this is more important, because these droplets, now you have all of these ApoE proteins together, if they're bound to amyloid beta monomers, each of them are bound to an amyloid beta monomer themselves, now there's a whole bunch of amyloid beta monomers coming together too, right? And because these amyloid beta monomers are coming together physically, that increases the chance that they may start forming oligomers. And they may start binding to each other and forming these multi-peptide oligomers inside of the brain. So lipidation of ApoE will increase the production of the oligomers. And last but not least, there is yet another mechanism that makes matters even worse, and that is there exists some proteases that can cleave ApoE in a manner that will make the ApoE start forming toxic amyloid beta oligomers much more quickly. Right? So this is proteolytic cleavage that can take place and will induce the formation of these oligomers. What proteases can do this? Two proteases that are known to cleave ApoE are the MMPs, the matrix metalloproteases, and another protease called calpain. So in this module of the network, I'm saying that these two proteases will induce the formation of truncated ApoE and, of course, reduce the levels of normal ApoE because they're cleaving normal ApoE. So when these two are activated, there's going to be a less ApoE and more truncated ApoE, and truncated ApoE is what can eventually lead to the formation of plaques. And the formation of plaques now can start interfering, not so much with neuronal function. We saw already that neuronal dysfunction happens earlier. But now it can start messing up with brain blood vessels, can bring in more inflammatory cells. So it's just a problem that keeps worsening. Okay? What activates these things? Well, among other things, that inflammatory state can activate these extracellular proteases. Often, inflammatory states induce the production of these things because it's like, we're in trouble, something's wrong. Just in case, let's produce all of these proteases and dump them on the outside because who knows what we're facing. And next thing you know, these proteases are breaking ApoE into truncated ApoE, making things worse. All right, we're getting there. This is my last slide. All of those bits that you saw before, all compile here. And this is going to be the starting points. Who knows what this network is going to look by the end of the semester.